for which I would like to invite Dr. Bichu sir, Principal, MES Asmabi. Sir, please.
Thank you. Respected Chief Guest of the Day, Dr. P.M. Serendaj, Head of the Department of Chemistry, Dr. Ansar, Head of the Department of Biochemistry, Dr. Arich, participants from different parts of the country. Good morning, all of you. MES Asmabi College is an aided college affiliated to Calicut University, and we are running more than 26 programs now, which includes degree courses in aquaculture, botany, and different BO courses sanctioned by UGC. And this year, during this COVID pandemic period, we have conducted more than 40 webinars organized by different departments. And here in Asmabi College, the biotechnology and chemistry department are actually single faculty departments, but they are organizing today a webinar on materials in medicine. We know that in the developing world, needs of new materials, novel materials are needed for different applications, and a lot of research are going on in this area in different parts of the world. Especially in medicine, we know that nowadays different novel materials are discovered for various applications, mainly for artificial tissue, artificial bones, and different nanomaterials are used in this medical world. In addition to that, in medicine for drug delivery, we are using different materials, and for detection of cancers and different diseases, we are using materials like quantum dots. And treatments, we are using different new materials. Different biosensors are discovered. And anyway, in medicine, there are a lot of changes, or a lot of novel materials are discovering using various researchers. And in addition, even and also in other fields also, there are novel materials. In material science, a lot of researches are taking place, especially in energy storage, and in polymer science also. There are conducting polymers and different types of light materials, and even in superconductivity, there are a lot of research taking place. And I am not explaining the field of research, because we have two eminent resource persons today and tomorrow for delivering for taking the webinars. And I congratulate the Department of Chemistry and Biochemistry and the IQAC of MES Asmabi College for organizing this webinar. And I think now 168 participants are joined for this program. And we are getting a very good participation from different parts of the country. I once again welcome our chief guest and all the participants for the program. Thank you. Thank you very much. Have a nice day.
Thank you, Bichu sir. Now, I would like to invite Dr. Shafir, IQAC coordinator of our college, to deliver a few words about this. Sir, please.
Thank you, Ansar sir. Thank you. A warm good morning to one and all. Chemistry has great role in our daily life. In the current scenario of pandemic COVID-19, chemistry can do much for alleviate our suffering. We know almost all major nations have engaged to find a vaccine. That is the reason why IQAC and Departments of Chemistry and Biochemistry have decided to conduct an international webinar on materials in medicine, which is highly relevant. I hope this webinar would add to the existing knowledge. I wish this webinar for all success. Thank you.
Thank you, Sheffield, sir. Let me introduce our speaker, Chief Guest, Dr. Payam Zerintax. Zerintax is an eminent academic researcher. Payam obtained his PhD in polymer engineering from University of Tehran, Iran, in the year 2018. He has several years of research experience in material science. He has published more than 70 research articles in reputed journals and has several patents to his record. He is currently holding a position as postdoctoral researcher in Oklahoma State University, USA. Over to you, Dr. Pai.
Thank you so much, Dr. Ansar. Also, first of all, I want to thank you for your kind invitation and your hospitality. Can I share my screen and my presentation file?
Sir,
Can you hear me, Dr. Ansar?
yeah,
Uh,
now it's
I want. Okay. Can I share my PowerPoint with you to start the presentation?
Yes, please.
Okay. Can you see my screen?
Yes, sir. Yes, yes.
Okay. First of all,
Yes, sir.
I want. Okay.
But sir, your volume is too low to hear.
Okay. Can you hear me?
Now it is audible, sir. Thank you.
Okay.
Now it's.
Can I start?
Yeah, sure, please.
Yes,
First of all, I want to thank you for your kind invitation. Today, I want to talk about the polymers application in the medicine. As you know, polymers have a different application in medicine and biomedical engineering, including tissue engineering, delivery systems, neural electrode, dental composite, and implants. Polymers can be categorized as a natural and synthetic polymers. Biopolymers can be achieved from natural source such as alginate, cellulose, and starch, or can be achieved from microorganisms like polyhydroxyalkanoids, or from biotechnology, polylactic acid, and finally can achieve from petrochemical products such as polycaprolactone. Polymer can be used in different parts of the medicine, and I can say it's the endless uses of polymer in the medicine because polymers are biocompatible and easy to produce. Usually they are cost effective and they can be designed to mimic the body and tissue. And also they can be used in a minimally invasive manner. Tissue engineering as one of the main important category of the biomedical engineering has several factors such as biological factors, cells, and scaffolds. Usually, biological factors and cells are embedded in the scaffold, and these scaffolds are placed in the injured tissue to regenerate the tissue properly. The scaffold is the engineered biomaterials to interact with the cells. Also, these scaffolds provide the proper environment to cellular activity and cellular growth. These scaffolds should mimic, or better to say, it's better to mimic the tissue properties. Polymers are the most commonly used scaffold in tissue engineering because they are biocompatible and they have the adjustable properties, and they also can mimic the ECM behavior. It can called ECM is the abbreviation of the extracellular matrix. As I told, the polymeric scaffold should mimic the tissue behavior to have the best and optimum regeneration. For example, brain tissue have the modulus around 1000 Pascal, and bone tissue has the modulus around 10 to the power 11 modulus. We use the hydrogel for brain tissue regeneration, and also we use the inorganic materials for bone tissue regeneration. Polymeric scaffold can be synthesized in different type. For example, they can be synthesized as a hydrogel, nanofiber, elastomer. They can be synthesized as a process structure and injectable hydrogel. For instance, I want to talk about the nerve regeneration, and I will tell you about my recent studies that we have done in neural tissue engineering. Neural injuries can be occurred by different factors, for example, vehicle crashes, violence, and sports. As you know, neural cells are composed of cell body, axons, and the myelin shells that conduct the bioelectricity through the axon. Axon damage can be categorized in compressed one to completely degeneration. When the nerve completely degenerate and cut it in two parts, various strategies have been used for nerve regeneration. The most the most commonly used strategy for regeneration for neural peripheral nerve regeneration is using the conduits. Conduits or guidance nerve tube are a tube that facilitate the nerve regeneration. They are sutures to the two separated part of the nerve, which is called proximal and distals. These conduits facilitate the nerve regeneration and connected these two separated parts. The gold standard method for neural regeneration is using the allograft and autograft, but they have some limitations such as limit of donor. But scientists, to overcome these limitation, they synthesize some synthetic materials. The first generation is silicons. Silicons are the biocompatible materials which can be used as a conduit, but these silicon-based conduit have some limitation.
Sir, sorry for interfering. Can you increase the volume? Your voice is little bit low.
If you let me, I try to increase its volume. Is it good enough?
Yeah, now it is good enough, sir.
The silicon-based conduits are not biodegradable, and we need the second surgery to remove the silicon-based conduits from the body, because remaining conduit in the body cause inflammation. Because of that, the second surgery is should be done for remove the silicon. Scientists to solve this problem, they use the biodegradable polymer such as polylactic acid and PLGA. In these cases, we don't need the second surgery to remove the conduit from the body, and they can be degraded within the body. But by increasing the distance between the distal and proximal, two separated parts of the nerve cannot spontaneously regenerate themselves, and they need some stimulations. Because of that, scientists designed the bioconductive polymers to increase the cell and nerve regenerations. Different strategies have been used for neural regeneration, such as using the growth factor, using the cells, and also using the electricity that I talk about the electrical activity here. As you can see in the figure, in the right panel is a polycaprolactone is a non-conductive substrate, and in the right panel is non-conductive substrate, and in left panel is the conductive substrate. As you see, the conductive substrate causes the increase in the proliferation and growth of the cells, and the electrical stimulation cause the morphology of the cells tend to the neural cells. The conductive materials have been used in scaffolds are metal nanoparticles. Conductive polymers are carbon families. The conductive polymers, the most famous conductive polymers that have been used in neural tissue engineering are polyacetylene polypyrrole, polythiophene, and polyaniline. Conductive polymers have wide range of the conductivity that varies from insulator to the metallic, and the electroactive polymeric composite varies from the semiconductor to the conductors. The dopant presence in the conducting polymers determine the conductivity of the polymer. If we add the conductive segment to the main backbone of the polymer, we show different properties from this synthetic polymer. Because of the difference in hydrophobicity of the conductive polymer and the biopolymers, we can see the self-assembly of the polymer, and we can see the different morphology. Also, the segment lengths of the conductive polymer and the end groups can affect the cell viability, and also the concentration of the conductive polymer affect the physical and electrical properties of the cells. The conductive polymers, because of the hydrophobic nature, they tend to self-assemble in biological environment, and they form like the sphere. Also, when we culture the cells on the conductive substrate, by the variation from the conductivity, cells morphology has been changed, and in insulating substrate, the cells tend to show the more proliferation, and in conductive substrate they want to change their morphology. As I told, dopants play important role in conductive polymer conductivity. You can see the different dopant conductivity here and its effect on cellular activity. Dopants have some problem because they are the small molecules. They are usually migrated from the conductive polymer, and this migration cause the polymer reduce its conductivity. To overcome this problem, scientists use the self-doped conductive polymer, and also they use macromolecules with acidic group as a dopant, like hyaluronic acid. We have lots of work in this area. We use the biopolymers such as chitosan, agarose, and alginate, and use this conductive segments such as oligoaniline to fabricate the bioconductive polymers. We use these conductive polymers, and we evaluate their conductivity. As you can see, we achieve the different and wide range of the conductivity here. Also, because of the rigid nature of the conductive segments, it causes the strength of the synthetic hydrogel will increase. As you can see, by adding more concentration, the strength of the hydrogel was increased. When we add the conductive segment to our hydrogel, as you can see in the left panel, the morphology is look like the honeycomb, and by adding the conductive segment, which is showed in the right side, the morphology change to the aligned one. It's because of the hydrophobic structure of the oligoaniline segments, because when they are encountered with the biological environment, they don't want to interact. They want to minimize their interact with the biologic environment, and the aligned structure was formed. The other important factors in in scaffolding tissue engineering are swelling behavior and degradation. As you see in this figure, The left panel showing the swelling behavior of the hydrogel with and without the conductive segment. Conductive segment, because of its hydrophobic nature, repel the water molecules and do not allow the water molecules to penetrate within the hydrogel and reduce the swelling ratio of the hydrogel. Because of this phenomenon, the degradation rate of the hydrogel was increased because of the presence of the conductive polymers. Based on this, we can adjust the swelling and degradation behavior of the polymer using the conductive polymers. Also, we can control the drug release behavior that I will talk about this slide in the other slides. Also, when we use the conductive polymer, we should use the optimum percentage of the conductive substrate, because conductive polymers in higher concentration shows the cytotoxicity, and this cytotoxicity cause the cell death. Because of that, we should use the optimum percentage of the conductive segments. With using the optimum conductive segment, we can achieve the maximum proliferation and better neural differentiation. After that, we use this conductive scaffold and fabricated the conduits. We cut the sciatic nerve of the rat, and we suture these conductive conduits to the distal and proximal. And after that, we trace the pattern of walking the mice, and we evaluated the nerve regeneration. Based on SFI, or sciatic function index, we concluded that the conductive substrate showed a better regeneration rather than the non-conductive substrate. The electrophysiological study revealed that the conductive substrate cause the nerve exhibit the electrophysiologic response from the beginning, and after 12 weeks we saw the proper electrophysiological performance of the sciatic nerve. In another work, we use the thermosensitive polymer poloxamer. We use the chitosan as a biocompatible polymer and aniline pentamer as a conductive segment for regenerate the hippocampus of the rat model. Hippocampus is a part of the brain that responsible for learning and some behavior of the rat. In this slide, you can see the regeneration of the hippocampus area of the rat, and the hydrogel along with the growth factor has a better regeneration compared to the other one. The other tissue that used in tissue engineering are cardiac tissue engineering. The scaffold that have been used for these scaffold should be electroactive, exhibit the proper elasticity and proper anisotropy, because the cardiac tissue have some action potential activity, and because of beating, the scaffold should be elastic and anisotropic because of the cellular formation on the scaffold. The elastic scaffold, the most famous elastic scaffold that have been used in cardiac tissue engineering, they are polyurethane and polyglycerol seboside. As you know, the polyurethane have the soft domain and hard domain, and these soft and hard domains responsible for some elasticity. The other usage of the polymer is delivery system. When we use the drug, we use the, for example, we use the pill every 8 hours. To avoid this thing, we use the polymeric carrier to maintain the drug concentration in the effective area. Usually, we use the stimulo-responsive platform to deliver the drug for the desired and targeted area. For example, the cancer temperature are higher than the other cells, and we can use the temperature responsive material to release the drug on the cancer cells. The most famous temperature responsive polymers are ploxamer and polyisopropyl Ploxamer is a tri-block copolymer that have the two hydrophilic segment and one hydrophobic segment. By elevating the temperature, the molecules tend to self-assemble. And after increasing the concentration, after the critical micelle concentration, they tend to form the gel. Polynipam show the LCSD behavior. This is the liquid in the room temperature, and after the LCSD temperature, by elevating the temperature, it tend to solidify. Chitosan is one of the most favorite polymers in tissue engineering, but chitosan is not a thermosensitive polymer. By adding the beta-glycerol phosphate to the chitosan, we can endow the thermosensitivity to the chitosan and achieve the thermoresponsive properties from the chitosan. Polylysin is the other polymer that exhibit pH responsive properties. If we want to release the special drug in the colon, it is recommended that use the polylysin, because polylysin are not soluble in lower pH. For example, when we encapsulated the drug within the polylysin, the drug can be keep untouched until reach to the colon, because the stomach pH is around 1.9 and small intestine pH is around 5. When these carrier reach to the colon and its pH is around the neutral, the drug can be released from the polylysin. The other responsive thing is electroresponsive materials. As I told in the tissue engineering part, if we have the conductive segment in our polymer and we synthesize the copolymer based on the conductive polymers, we can load the hydrophobic drug within these cells, within these carriers. Because the hydrophobic drugs can interact with hydrophobic segments, and these hydrophobic segment can entrap the hydrophobic drugs. By applying the electrical stimulation, these drugs can be released within the body with arbitrary manner. The other work that we have done in our lab is using the protein delivery for cancer cells. We encapsulate the glucose oxidase within the polylysin and polyethylene glycol, and we use them for intracellular delivery of the GOX. GOX or glucose oxidase is a protein that use the glucose and produce the hydrogen peroxide. Glucose oxidase treat or kill the cancer cells in with two mechanisms: the starvation mechanism and the ROS generation. Glucose oxidase use the glucose and use the feed of the cancer cell nutrition, and because of that cancer cells don't have the enough feed to nutrition and cause the killing the cancer cell. By producing the hydrogen peroxide as a ROS or reactive oxygen species, these ROS can attack to the cancer cells and kill the cells. In the other work, we fabricate some neural electrodes to record the brain activity. We have a different type of the electrodes to record the neural signals, or some electrodes used to stimulation of some parts of the brain for some in some diseases. We synthesize the polyaniline and we fabricate in the nanostructure, and we can record the EEG signals of the brain, which is used for brain studies. The other composite, the other polymeric materials that have been used in the dental composite are based on acrylate resins. That they are the usually epoxies that they can be polymerized using the ultraviolet and can fill the tooth cavity using this polymerization. And finally, the other stage of the polymer, I can say that polyethylene terephthalate can be for vascular graft, or polymethyl methacrylate can be used for the contact lens, or the polyethylene can be used for knee replacement, and some other materials such as silicones for face implants and polyethylenes for balloon catheter and other implants. In conclusion, I can say that polymers have a wide range of application in medicine, because polymers has the tunable properties, proper compatibility, and adjustable degradability. And it is open the polymer usage in medicine. And finally, I'm the guest editor of the Polymer Journal, and I will happy if you submit your manuscript within these journals. And finally, thanks for your kind attention.
Thank you, Dr. Payam.
You're
Now the
welcome.
session is open for a discussion. There are a few questions from the audience. I will be read. Please note that one. First one is, which is the metal and biopolymer suitable for the preparation of implants?
Sorry, would you mind repeat your questions? Your voice has interrupted. I cannot hear your voice properly.
Okay, then I will read one more time. Which is the metal and biopolymer suitable for preparation of implants?
The biopolymers are suitable for implants. We have the different kind of the implants. As I told, we can use some. We can synthesize some contact lens or knee joints. For example, knee joints, they are use the polyethylene, but they are the synthetic polymer. The biopolymer we can refer as some synthetic and natural one. In natural one, they are usually degradable, and in implants, we want to non-degradable polymers, and we usually use the synthetic polymer. For example, polyethylene, ultra-high molecular polyethylene is the best choice for using the knee implants. Or we can use the polymethyl methacrylate as a contact lens. Or we can use the polyethylene as a catheter for cardiac vessels.
Okay, thank you. There is another question. Silicon has good biocompatibility.
Yes.
Is it right or not? Something like that. It shows they are putting some question marks. I think they are asking maybe silicon has a good biocompatibility, or what is the importance maybe?
Silicon is a biocompatible polymer, but we should we should use the silicon properly. If we use the silicon in as a nerve conduit, we need the second surgery to remove the silicon. And it's not recommended to use in the body because it's not degradable. We can use it in some usages that we want to non-degradable. We can synthesize some vascular based on the silicon, or we can synthesize some wound dressings based on the silicon because they have the proper performance on wound healing, and we can use in the wound healing applications.
There is one more question. Which polymer used in current medical treatment? Which biopolymer used in current medical treatment?
Would you mind say it again?
Which biopolymer used in current biomedical application? I think so.
Which polymers used in biomedical application? Am I right?
Biomedical treatment.
We have a different kind of. It's based on our purpose. We can use different kind of biopolymers for biomedical application. For example, if you want the thermosensitive polymer, you can use for example, biopolymer. You can use the chitosan with polyglycerol with beta-glycerol phosphate, and or maybe. If you want to drug delivery, you can synthesize them as a nanoparticle, or you can use the chitosan alginate, and these polysaccharides are the best. But for tissue regeneration, it's recommended to use the collagen and gelatin, and these type of polysaccharides like chitosan are proper for this usage.
Is these polymers are currently using in medical treatment?
About what?
Any other diseases or drug delivery application?
They have the wide variety of applications, such as tissue engineering light. It can be used in nerve tissue engineering, cardiac tissue engineering, or maybe in used as a wound dressing and bone tissue engineering. And it can be used in the drug delivery, for example, as a pH responsive and thermoresponsive.
Another question. Can you tell me the name of biopolymer or polymer used in medical sutures?
Used in the
medical sutures.
In the sutures, if we want to the degradable one, we can use the PLGA, polylactic acid, and these type of polymer are so proper for using in the suture. For example, polylactic acid is one of the main polymer that's used in suture, but it has some drawbacks, such as its brittleness, and you can copolymerize with other polymers to overcome this draw. And for example, you can use PLGA for your biodegradable sutures.
Another question, which polymer is used in a drug delivery application? I think current medical treatment.
In delivery application, you should consider if you want to use the polymers as delivery application, want to inject within the body, and it circulate within the body, you should consider the protein corona formation around the polymer. You should use the hydrophilic polymer and the Zwitter ionic groups to avoid the protein corona formation around the polymer carrier to deliver the drug to the desired to the target, and release the drug on the desired area. It's better to use the superhydrophilic materials, such as polyethylene glycol and or Zwitter ionic materials.
Okay. But there is a connected question, I think, previous one. Which polymeric nanomaterial, nanoparticles is better for cancer drug delivery?
It's depends on your purpose. It's for cancer treatment. As I told, it's if you want to use, if you want to inject it and deliver the drug to the cancer, for example, doxorubicin is an anti-cancer drug that have been encapsulated within some polymers, and it can be used for cancer treatment. The cancer pH and the cancer temperature are different from the normal cell. You can use the temperature responsive and pH resistive carrier, pH responsive polymer to carry the drug to the cancer. For example, you can use the polyene isopropyl acrylamide as a thermoresponsive polymer and polylysine as a pH responsive polymer for deliver the drug to the cancers.
There is another question. What are the troubleshooting come across during the delivery of polymers?
The main problem in delivery system, as I told, is the protein corona. Protein corona are the proteins that are available in the body, such as fibrinogen or albumin, that interact with the polymeric nanoparticle, and they can eliminate it by the immune system of the body. If we want to reduce this interaction of the polymeric materials with the proteins that are formed within the body, we should use the superhydrophobic material, hydrophilic materials such as polyethylene glycol or Zwitter ionic materials. If we add the Zwitter ionic groups on our polymer, we can create the proper hydrophilicity and prevent the purinocoretin formation. And we can troubleshoot of the elimination of the nano carriers within the body. You are muted, Dr. Ansar. Dr. Ansar, you are muted. I can't hear you.
Okay. Can you hear me?
Yes, I can hear you.
There's one more question. Can plant-based polymers using anthocyanins and tannins be used in medical application?
What kind of polymer that you mentioned?
Polymers used anthocyanin, plant-based polymers, anthocyanins and tannins be used in medical applications.
Actually, I don't know about that.
Okay. There is one more question. Any biomaterial being developed for more replacing more delicate tissues like eardrum and ear ossicles? Any biomaterial being developed for more replacing more delicate tissues like eardrum and ear ossicles?
Would you mind? I have some interruption in your voice, and I don't really understand your questions.
Okay, then materials is using for replacing delicate tissues like eardrum and ear ossicles. Eardrum and ear ossicles. Any materials is used.
The materials that have been used also, they use the biopolymers usually that we used. The main polymers that we used also are chitosanes and other polysaccharides.
Okay, polysaccharides is possible to use. Uh, okay, if there are any questions from participants, you can unmute your microphone uh, and you can ask directly to Dr. Pai. Participants, please. There is one more question. Nano missiles. Nano missiles versus nanoparticles. Which one is better for drug delivery system?
It's depend on your final usage. It's if you want to deliver. It depends on the drug and the final applications. If you want to embed the high dosage of the drug, you can use the nanomicelles, and you can use the nanoparticle for interacting with the drug to deliver the drug to the desired area. It's based on your target. It's completely have the it's a need very profound discussion to answer this question. For example, we embedded we use the nanoparticle, we use the polylysine and polyethylene glycol. We use it for deliver the GOX to the cancer cells. We use the electrostatic interaction between them because the GOX has the negative charge and the polylysine have the positive charge, and because of that we can synthesize the nanoparticle that delivers these combination to the cancer cells. And also the nanomicelle, you can use the ploxamer to synthesize the nanomicelle. You can load the hydrophobic drugs within it to deliver the hydrophobic to the desired target.
Okay. There's one more question. Do anti-inflammatory drugs have some curative properties for COVID?
About would you mind to say ask your question again?
Do anti-inflammatory drugs have some curative properties for COVID?
Actually, for COVID, anti-inflammatory, I'm not a pharmacist. I'm a polymer engineering. We just use the dexamethasone as an anti-inflammatory drug for our research in, for example, neural tissue engineering to avoid the inflammation in when we suture the conduit to the nerve. Actually, I don't have the enough information about the anti-inflammatory drug.
So good morning, sir. This is Shamina from Coimbatore. Actually, I have raised this question like, do anti-inflammatory drug has the coincidence with COVID? The thing is that when I worked with bioinformatics target for a drug, I saw a few drugs coinciding with the COVID-19 also. That is why when I write a paper, I have to be a little clear. That's why I asked this question to you, sir.
As I told, I'm not a pharmacist and I don't have a profound knowledge about the anti-inflammation drug, also about the COVID. I think the person who have the enough knowledge and or pharmacist can ask this question.
Okay, sir. Thank you.
You're welcome.
There is one more question. What buffers can be used for maintaining the pH of extracellular matrix during cell culture?
PBS is the proper one that we used for during the cell culture, I think it's useful.
Phosphate
Yes.
buffer solution.
Yes.
I think if there are any questions, participants can directly ask to Dr. Pai.
Dr. Payal, this is Vaishnavi, and it was a very wonderful presentation. I would like to ask a question regarding the source of polymer that was used for experimentation in mice. Is it natural or is it synthesized in lab? Can you please share about it?
We synthesize different type of polymer. We also use the natural polymers. For example, we use the chitosan, we use alginate, we use gelatin. And these natural polymers, we combine them with conductive polymers, conductive segments. For example, we use the different segments of the aniline oligomers, for example, the aniline pentamer, aniline tetramer, and aniline dimer. We use the aniline pentamer for neural regeneration. We use the combination of the agarose and aniline pentamer for regenerate the peripheral nerve, and also we use the chitosan and aniline pentamer for hippocampus regeneration along with the growth factor VEGF. These kind of polysaccharides we used for neural regeneration.
Thank you.
You're welcome.
Any more questions, please? If there are no more questions, we have come to the closure session. Before winding up, I would like to extend my sincere thanks to Dr. Payam for being such an excellent lectures on this potentially relevant topic. I would also like to thank the audience for their patient listening and participation. Participants, please note that a feedback form is sharing in chat box on last day. That is on 5th October. That is the second day of our webinar. Please make your response in that form. It will be sent to you the registered email address. Thank you once again. Thank you all.